So in the last class, we found that there are 3 or 4 forces that affect how the microevolution happens, and among them selection is the only one that is capable of explaining the process of adaptation. In general, selection is supposed to be a very strong force of microevolution, and in this particular topic we are going to look at some of the concepts related to this related to selection. So how does one define selection? There are many ways of doing it. One of the most common ways is what is taken from this book by Zimmer and Emlen. So natural selection is the differential survival and reproduction of individuals due to differences in phenotype. So the basic point over here, as we discussed, is that there needs to be differences across individuals in terms of a certain phenotype. That phenotypical difference should lead to a difference in how many babies they make, and this difference in phenotypes, at least partially, should be inheritable. So these are the 3 major conditions. Now, the distinction between natural and artificial selection, it's not very important, in the sense that in the literature the artificial selection is typically used for those selection pressures that are imposed by human agency, whereas pretty much everything else is supposed to be selection by nature and hence natural selection. There isn't any difference in the way in which these 2 kinds of selection operate, and therefore many modern books don't really make this distinction anymore. So before we start, we are going to look at one video on how selection works both on the short as well as the long time scale in the Galapagos finches. So the work that I'm going to show you was done by Peter Grant and Rosemary Grant, and this is one of the most famous pieces of work in evolution in the 20th century. So this particular video is from HHMI page, and this not only contains the video, this page, it also contains some extremely fantastic activities. So I really recommend that you guys have a look at those activities. At the same time, we are going to look at one chapter from Freeman and Heron, which will give a more rigorous account of this study compared to what is presented in the video. So even if you read the video, I strongly recommend that you should read the chapter. And finally, there is this fabulous book called The Beak of the Finch by Jonathan Weiner. It's a Pulitzer Prize winning book, as you can see. And it's one of the best non-technical introduction to the work on the Galapagos finches over the last actually now it's more than 40 years, but the book I think looks at the first 20-25 years or so. So let's have a look at the video. So there are actually 2 videos. The first video is about the main work over there, documenting how the Grants went about documenting natural selection in nature. And the second video is one of their more recent works, where they ended up showing the process of speciation in those islands, happening over a very short time scale. So here are the videos.
Our planet has millions of species, over 300,000 beetles alone, 17,000 butterflies, thousands of mammals, fish, and birds, all astonishingly different. How did so many species come to be? To seek insights into that question, researchers are focusing on places where species recently arose, such as the remote Galapagos Islands.
Scientists are making observations and conducting experiments that would have surprised Charles Darwin. And they're discovering new insights into what the great naturalist called the mystery of mysteries. how new species form.
The Galapagos Islands are one of the most spectacular landscapes in the world, home to a variety of species that live nowhere else. Biologists Peter and Rosemary Grant have been seeking answers to how species arise by focusing on one of the smaller islands called Daphne Major.
When we started out, we had no plan for the long term. In fact, we thought it was just going to be just a few years, maybe 2 years.
2 years have turned into a 40-year odyssey. The Grants have returned every summer since 1973.
Oh, that's a bird.
Is that 3 or 6?
That's 3 metal 6.
Here, they've made some of the most remarkable observations in the history of field research as they studied the famed Galapagos finches. The finches were first brought to scientists' attention by Charles Darwin when his voyage around South America brought him to this cluster of islands 600 miles from mainland Ecuador. These volcanic islands are geologically young. They began rising from the ocean floor less than 5 million years ago. At first devoid of life, they now support a modest number of species. Among them, 13 species of finches, found in various combinations on the different islands. The birds live in diverse habitats.
The islands are very different from each other. They differ in size, they differ in topography, and in height.
Larger trees grow at higher elevations, while low islands have mostly cactus, grasses, and shrubs. In these diverse habitats, the finches have evolved many ways to survive.
So, Rosemary, what's the important difference between these birds?
This little warbler finch with its very fine needle-like beak is perfect for picking off insects. This one is the woodpecker finch with a rather more robust beak. It concentrates on beetle larvae and termite larvae. Then we have the cactus finch with a much longer, sharp-pointed beak, which probes into cactus flowers. And then these 3 species are the large, medium, and small ground finches. So, Sean, the basic idea is the beaks are tools, and you need the right tool for the right job.
The finches look so different that Darwin first mistook them for entirely unrelated kinds of birds. How did the Galapagos end up with so many species of finches?
In terms of the actual history of the finches of the Galapagos, there were many different possibilities. Different kinds of finches could have all come from the mainland separately, or the finches could have all evolved out there on the islands. And what do we know about that?
Well, now we know from DNA evidence that all of the finches are more related to each other than any one is to a species on the mainland. And that tells us only one species arrived on the archipelago and diversified into the 13 species that we see nowadays in the Galapagos. So they've all come from a single common ancestor.
The question then becomes, how did one ancestral population give rise to many different species, each adapted to a different lifestyle? A crucial insight into how adaptation occurs came when the Grants focused on one species on the island of Daphne Major.
factor of great convenience for us was the small size of the island. That meant that we could walk all over the place.
Oh, there's a bird.
I'll leave that one for you.
The idea was that if we worked really hard, we could follow every individual or almost every individual.
They rose at 5:30 each morning to net the island's medium ground finches.
Beak net is...
They measured the size and shape of each bird's beak, the bird's weight, and they tagged them for identification.
The male is 17418.
Year after year they returned, at times tracking over 1,000 finches.
So here's an example of a bird we know intimately over the whole of its lifespan. The number is 5960. We know how many times it bred, which years it bred in, how many mates it had, how many offspring it produced, and then how many of those offspring themselves survived long enough to breed.
Over the first 4 years, little seemed to change. Then in 1977, a terrible drought began.
Virtually no rain fell for the next 18 months.
The vegetation practically disappeared, apart from a few trees without any leaves, and of course the cactus bushes were still there.
Now the medium ground finches had to compete for scarce food.
They started off with a big food supply of small seeds, medium seeds, large seeds. As these small seeds became very scarce, they had to turn increasingly to the large and hard seeds. Well, only birds with large beaks can crack open these woody, spiny fruits.
The birds with the smallest beaks had the most trouble.
They were scraping about amongst the rocks, and their plumage got so worn that they could barely fly.
That year, over 80% of the medium ground finches died.
We would go around looking for birds that had died, and it's very sad to pick up a bird and say, 3972. Oh, no, not that bird. Oh.
When they inventoried the surviving medium ground finches, they discovered that one trait had made the greatest difference between life and death.
Well, I'm showing here a distribution of beak depths of the population in 1976. The survivors of this group are shown in black.
Oh. So the larger the beak, the better your chances.
The larger the beak, the higher the likelihood of surviving through the drought of 1977. 18.6 grams.
When they looked at the offspring, they found an even greater surprise. The average beak depth was more than 4% larger than the previous generation. Natural selection had changed the average beak size.
Could you have ever imagined measuring and observing something like this on such a short time scale until you actually did it?
When we started, the answer is no. We could not imagine we would be able to do it.
But was this a fluke? Or are changes like this happening all the time? 5 years later, in 1983, an unusually strong El Nino brought 10 times more rain than normal. And the island was overrun by vines that covered even the cactus. The rains changed the vegetation on the island, such that 2 years later, when drought struck, larger seeds became scarce. The birds with larger beaks now had difficulty picking up the more abundant food, the small seeds produced by the vines. That year, many more finches with small beaks survived, and their offspring inherited smaller beaks.
So the selection had swung in the opposite direction, and evolution had occurred as a result.
In an amazingly short period of time, the Grants had measured evolution of beak size not once, but twice, demonstrating that when birds encounter different environments,
They will change over a very short amount of time. Over millions of years, changes like these, occurring throughout the Galapagos, generated all sorts of beak sizes and shapes. But that's only part of the story. How did finches with different beaks become distinct species? Species are defined as populations whose members don't interbreed.
So how does one species split into two? A typical scenario is that two populations become separated geographically and undergo enough change in their respective habitats that if or when they come into contact again, they do not mate.
So in the Galapagos, the Grants asked, what keeps different species of finches from mating?
We were very conscious that on any given island, the different species sing very different songs. This is what a cactus finch sounds like. Whereas the medium ground finch sounds very much like this.
So to see if songs keep the species apart, the Grants and their student Laureen Ratcliffe played each species' songs through a loudspeaker.
When we played back the cactus finch song, cactus finch came to the loudspeaker and the medium ground finch completely ignored it.
The males only responded to songs of their own species. The Grants looked at whether finches might also choose mates based on appearance. So they put out stuffed female specimens to see if males would respond.
Clearly they could discriminate. The male vigorously courted a female of his own species, completely ignored the other one.
The males only courted females that had a similar size and similar beak. Song and appearance both play a role in keeping different species from mating. So when populations of the same species are separated, changes in these traits set the stage for the formation of new species. The Grants have shown that both geography and ecology are keys to the evolution of the Galapagos finches. The most likely scenario is that 2 million years ago, a single finch population arrived from the mainland. When their descendants reached another island, they faced new conditions. As those isolated populations adapted to their surroundings, their traits changed. If the changes included traits involved in mating, and the populations came into contact again, they no longer mated. They had become distinct species. While unique to these remote islands, the history of the Galapagos finches offers a general insight into why the world is populated with so many species.
The more diverse the environment, the more opportunities for evolutionary change to produce those new species.
Over 150 years after Darwin first recognized their significance, these unassuming birds still illuminate how the great diversity of life arose and continues to evolve.
How does a new species form? The standard explanation is that the process begins with part of a population splitting off and entering a new environment. In a paper published in Science in November 2017, we show how hybridization can play a role. This bird is a member of a new lineage of finches on the Galapagos island of Daphne major. The lineage originated by hybridization of an immigrant with a resident species. The immigrant was an exceptionally large male with an unusual song. To our surprise, DNA sequencing from a small drop of its blood revealed that it was a Conirostris, like this one, from Hispaniola Island 100 kilometers away. The immigrant eventually bred with a resident Fortis, like this one. Their offspring learnt their father's unique song and bred with each other, and so did their offspring. We followed this highly inbred lineage for 6 generations over more than 30 years. Interestingly, members of this lineage differ from both their Conirostris and their Fortis ancestors. They have large beaks relative to their body size, and they eat a wide variety of foods. The key to their ecological success and reproductive isolation is their unique morphology and distinctive song that differs from the other 3 finch species on the island. 3 generations of big birds, Fortis, Scandens, Magnarostris. The lineage is flourishing, but how long it will survive we do not know. Regardless of whether the new lineage succeeds or not, it provides an example of one way in which a new species can arise through hybridization.
So, to put the whole thing in a nutshell, if you have a breeding group, a population, and in that breeding group, there is variation among individuals in the context of a given trait, if at least part of that observed variation is heritable to the offspring, if some of the individuals are more successful in reproduction, that is, they have greater fitness than the others, and if the phenotypic variation that we talked about in one has a direct effect on this this variation in terms of reproduction, then if all these 4 conditions are met, then the phenotypes which have the capacity to leave behind more babies, they are the ones whose frequency in the population increases, and they are the ones who are supposed to be quote unquote naturally selected. As I said, naturally, the word naturally is not important over here. The word selected is the one where you should concentrate. Now, what we will do is we'll take the Darwin's finches case study that you saw in the HHMI video, but we are going to look at it in a greater detail, explicitly seeing whether these 4 conditions are being met or not, and if they are being met, then how does one make inferences about how selection is operating in a system. So as I just said, that this is 3 decades of research by the Grant couple, and the Galapagos Islands, they in some sense are a natural laboratory because you have multiple islands which have roughly similar conditions, not entirely but roughly, and since these are small islands, therefore one can actually go around and tag every single bird and track them across generations, which is what the Grants did. And of course it's relatively simpler to monitor the survival, reproductive success, and the beak size, beak shape, etc., on site without really bringing them to the lab. So, as you know that these islands lie about 800 to 1100 kilometers west of Ecuador. And the study that we are going to talk about concentrates on this particular island called Daphne Major, which in spite of the major in its name is actually a pretty small island. So apparently if you start at any one point on the rim and simply walk along this rim, you can actually come back to this point in about 20 minutes. That's how small an island it is. So we know that these finches, they arose from one particular species of finch, which apparently molecular dating suggests that this flock migrated about 2.3 million years back from the mainland. And over these 2.3 million years, these birds have diverged quite a bit, so that now we can recognize about 14 species primarily based on their beak shapes and beak sizes. Now, remember what we are going to do over here is we are going to see whether those 4 conditions that I outlined, they are being met or not in the context of selection. So the first thing is that there has to be genetic variation and phenotypic variation for a trait related to fitness. And in the context of these birds, the trait that we are going to look at is beak size. Okay. Now, beak size is very important because it essentially dictates the success of these birds in terms of foraging. So what it is that they can eat, how easily they can handle it, depends on their beak size. So we are going to look at one particular species, Geospiza fortis. And if you look at the beak size or beak depth, as they call it over here, this is the beak depth. So if you look at the population of these birds in 1976, there were about 751 individuals, and you can see that there is a very nice distribution in terms of the beak sizes. So suggesting that there is phenotypic variation for this particular trait. Now remember, it's not enough to have phenotypic variation. At least part of this variation needs to be heritable. Now how will you test whether a part of it is heritable or not? So obviously, in order to check whether something is passing across from parents to offspring, you are simply going to look at the thing both in parents and in offspring. So you can either do it that way, or you can also measure it across siblings. That also gives you a way of doing it. So the response, if there is strong response to selection, or for strong response in selection to happen, this variation has to be heritable. So how do you measure it? So what you do is you take whatever is the trait that you are considering, and you measure it in both the male parent and the female parent. Obviously, you are going to get 2 values. So you take the arithmetic average of that, and that is what is called the mid-parent value. Now you take all the kids of this particular pair and measure the same trait across all of them, and take the average across all the offspring. That is what is the average offspring value, right? So in order to see whether this is influencing this or not, what you do is you measure a quantity called the correlation. What does it do? So without going into the formula, suppose you have a scenario which looks like this, where these are the parent trait values, these are the offspring trait values, and you can see that in general, as the parent trait value increases, the offspring trait value also tends to increase. So that means that if you know the parent's trait value, you can reasonably predict what will be the direction of the offspring's trait value. So if, for example, going from this parent to this parent, you can say that, okay, the trait value is going to increase. So this is what is meant by having a strong correlation, right? So whenever there is this kind of association, the correlation coefficient is supposed to be high. On the other hand, if you have a situation like this, where there isn't really a major pattern in terms of the parent values and the offspring values, then this is what the thing is going to look like roughly. And the main point to note is that if you fit a best fit line to this line is going to have a much shallower slope. It's going to be more flat, whereas in this case it will have a stronger slope. And this stuff in the literature is known as the heritability of the trait, right? And the greater the heritability, the stronger is the inheritance component, and vice versa. Now the problem is that what do you mean by greater and what do you mean by lesser? So roughly speaking, it has been shown that if the heritability value is greater than 0.2, then the trait can respond pretty fast to selection. Whereas if it is less than 0.2, the trait is going to be slow to respond to selection. This is just a thumb rule. Many people might even disagree with it, but this has been proposed in the literature. So just to give you an example, in the context of human height, the heritability is 0.6, which means that there is a very strong association between the parent's height and the offspring's height. So now the question is, what is the heritability over here? So when you measure when you plot the offspring value against the mid-parent value, these are the things that you get. So this is, the green ones are in 1978, the blue ones are in 1976, and in both years you can find that there is a reasonable relationship. As the offspring values, as the parent values increase, the offspring values also tend to increase, which suggests that the beak depth in finches are actually heritable. So the second condition is being met. So now the point is that whether whatever beak depth you are finding, that has something to do with the fitness, that has something to do with the survival and or reproduction of the individuals or not. Now that is something which actually is the tougher thing to show. However, the Grants got a little lucky in some sense. In 1977, there was a major drought on Daphne major, and the rainfall was less than 20% of the normal levels. When that happened, most of the plants failed to flower, and therefore seed production declined on the island. Now, as a result of that, the birds died in very large numbers. How did it go? So, beginning 1975 or so, mid-1975, the population size was about 1400. So roughly, it had started declining a little bit, but when the drought struck, this number really declined. And then by the time the drought was, at its full flow, 1978 was starting, the population size had fallen from about 1400 to about 200. Okay. So obviously, not everybody died. Some of them died. So which meant that there was a variation in terms of the individuals, in terms of their survival ability. Now the point was that what was causing this variation? So if you look at the abundance of seeds, you find that before the drought strikes, that our seed abundance is pretty high. It's about 10 or so grams per square meter on the island. But then as the drought starts, you can see that it actually falls drastically to about, say, 2 grams or so per square meter. So it falls about 5 times or so. Now, not only does the number of seed fall, but the characteristic of the seeds, they also change. So what they have over here on y-axis is some kind of a composite measure of seed characteristic. So as you go higher, this is the seeds are larger and harder, and as you go smaller, the seeds are smaller and softer. So if you see this, you see that right at the beginning of the drought, the seeds, an average seed, so a seed picked up in random, was going to be small and soft, whereas as the drought progresses, these small and soft seeds basically get eaten up, and therefore what are left are actually the larger and the harder seeds. So which is why the characteristic of an average seed shifts towards largeness and hardness. So the resource availability is changing drastically. Now when this is happening, what happens to the survivorship due to these changes in seed availability? So it turns out that there is a very nice pattern in the way the birds survive. It's not a random thing. So this is the distribution as we saw in 1976 before the drought strikes. However, if you look at the survivors in 1978, the distribution looks very, very different. If you see that although there has been a lot of death, the death has been more in terms of these classes. Okay, so basically the guys who had smaller beak depths, proportionately speaking, their numbers have fallen more, whereas proportionately speaking, those guys who had larger beaks, these guys, their fraction in the population has ended up increasing, right? Now, why exactly is this happening? This is happening because the seeds that the birds can eat, that depends on their beak shape and beak size. So those birds which have relatively smaller beak sizes, they are much better in terms of handling small and soft seeds. However, those birds which have larger beaks, they are not so good, not so adept at handling the smaller seeds, but they do much better in terms of handling the larger seeds. So when the seed production fell and the average seed characteristics moved from being small and soft to large and hard, then actually the birds which had the larger beak depths, they were favored, and therefore, although they also died, but their death was, at relatively speaking, at a lower rate compared to those guys who had smaller beaks. So we see that all the four postulates of Darwin that I showed you, all of them are being met. So now the question is, did the populations evolve? And if you look at, now remember, if you simply look at this and say the population is evolved, then it doesn't work. Because all that this is saying is that the larger beaked guys, they survived better. However, the real thing is that not that whether these guys survived more, but were they able to also pass on this trait to the next generation such that the frequency of these guys increased in the next generation or not. And for that, you actually need to look at the frequency distribution of the finches which hatched after the drought and compare them with the ones before the drought. So as you can see that if you look at the finches that hatched before the drought, the mean was somewhere over here, somewhere about 8.9 or something. Whereas if you look at the finches which hatched after the drought, the distribution had actually shifted to the right hand side, and therefore the mean of the beak size had increased some amount. I mean, this is statistically significant. So suggesting that there has been microevolution at the level of these Galapagos finches. So as I said, there was a major shift in 1977 towards the big and the deep beaks. Now, when there are moist years, then there are lots and lots of soft and small seeds. When that happens, then the selection actually moves in the reverse direction. Now, obviously the question over here is that if you have a big beak, then you should be able to eat both the big seeds and the small seeds. So why isn't selection making all the beaks big forever? That might be the case. We don't exactly know this, but based on whatever people have conjectured, that may be happening because it's possible that the bigger beaks aren't better under all the circumstances. So for example, the bigger beaks might be poorer at manipulating the smaller seeds. It might be energetically more expensive to produce bigger beaks because the growth is less, their energy for available for other functions, other body parts is less. This we know for sure happens, as we saw that in these birds, the vocal communication is a very, very important part because it affects their mate choice. And obviously when you increase the beak size, the kind of vocalizations that you will make is going to change. And also it is entirely possible that this is genetically correlated, beak size, with other traits under selection, which means that if you are having large beaks, then you might end up having genetic issues in terms of some other traits. The values of those traits and the fitness along those traits might go down. Now these are all conjectures. We don't really know which exact one of these is causing why, or preventing the evolution of big beaks forever. However, what we do know is that, as I said, when conditions change, then life becomes very interesting. So what we have over here on the x-axis is time in years, and what we have on y-axis is a principal component analysis of the beak size, which basically means they take multiple properties of the beak size and combine them into one index. So this is what is the scenario around early 1970s. So what this represents over here, this is the mean and this is the variation around that, and therefore this is in some sense the confidence interval around this mean. Now if you look at this over here, there is some amount of up down that happens due to genetic drift. These are small populations after all. This increase in beak size is the one that we discussed right now. This is the one that happened due to the drought. Once this happened, then it stayed like that for some time, and then the rainfall levels slowly became normal. The plants slowly came back, the seed production changed, and when that happened, again now the smaller beak guys were favored more and more. As a result, the average beak size started coming down. So then it again came down to its original levels, and then it bounced around a bit, and then over here this was an extremely moist year. Okay, so a lot of rainfall, and when that happened, then there was a superabundance of soft seeds, and then the selection pressure actually operated in a big way against the large beaked guys, and therefore this fell. Okay, this fell badly, and the beak sizes actually became much smaller. Now think about it. We have this kind of a data because the grants were following this year after year, and therefore we have the yearly means. Now suppose you do not really have that kind of a resolution. Suppose you have, let's say, 20-year resolution or 30-year resolution. Then what are you going to see? Suppose you have measured it here, and then you have again measured it, say, here, and you have measured it here. You are going to see, roughly speaking, the thing remaining at roughly the same level, the beak size remaining roughly in the same range. Now, this is just a few decades. Now, suppose you are seeing this over millions of years. What will happen? You are completely going to miss all these seesaws that are happening in the trait values. You are essentially going to see a large, nice, unbroken period of let's say 1 or 2 million years, during which there is hardly any change in the beak sizes, right? So why am I bringing this up? Because you remember when we were talking about modern synthesis, we said that there the idea was that evolution happens in a very, very slow pace, right? However, when we are looking at this, we can see that evolution is happening actually at a pretty fast pace, right? It's happening right in front of their eyes over a time span of just 1 or 2 years, and that too in birds. If you talk about microbes, evolution can happen, 15 days, 1 month, evolution, massive evolution of antibiotic resistance, etc. happens. So, evolution is fast or slow. That depends on what is it that you are tracking. If you are tracking microevolutionary changes, that is changes in trait distribution, then they can actually be very, very fast. They can happen over small time scales. However, if you are tracking macroevolutionary changes in, let's say, not the beak shape per se, but a new character coming in and so on, then those changes might, not necessarily, but mostly might end up taking very long periods of time. Now remember, many of these people who were talking about modern synthesis, they were also trying to think in terms of macroevolution. They are thinking they are basing their intuition on fossil data. And if you look at fossil data, which of necessity is happening at much coarser time scales, then these kind of nice seesaws, you don't really see them over there. What you see is a smear, and that is why people whose intuitions are coming from that side, in other words, people who are thinking about macroevolutionary changes, thinking in terms of fossil data, they end up thinking of evolution as a slow process, whereas people who think in terms of microevolutionary processes, they end up thinking about evolution as a fast process. There isn't really a major dichotomy between the two. Okay, so what we are going to do is we are going to now talk about a few major insights that are coming out of the Daphne major work. So, as I showed you, microevolution can occur very rapidly and very dramatically, and sources of natural selection, they end up shifting often. So as I showed, 1 year when there is a lot of drought, big beaks are favored. Another year when there is no drought, a lot of rainfall, small beaks are favored. So selection, the direction of selection itself ends up changing dependent on environmental changes, and when that happens, evolution itself can reverse its direction, or sometimes go in a completely new direction, new traits, and the organisms end up evolving a different way of solving the same problem. And as I said, this seesaw of selection may give the impression that you are having a gradual change, but that's only a time scale issue. And if you are trying to resolve this using fossil records, then you are typically going to miss this fast, up down in the evolutionary data, in the evolution trajectory. So, a few words about the nature of natural selection. So, selection, roughly speaking, is divided into three major types. So, suppose this is the distribution of the phenotypes, and only individuals in one tail of the distribution, this tail or this tail, these are the ones who have greater fitness, and these are the ones who have less fitness or zero fitness. Then essentially, the mean of the distribution is going to continuously shift in one direction. So this is what is known as directional selection. On the other hand, you might have a scenario wherein, let's say, individuals who are either too small or too big, which basically means which are on either tail of the distribution, both of them have low fitness, whereas those individuals which are in the middle of the trait distribution, they are the ones who have higher fitness. So this is something that happens very often with traits like, traits which are related to the physiology. So for example, body weight. So if you are malnourished and underweight, then your fitness is very low. If you are obese and grossly overweight, then also your fitness is very low. Whereas if you are somewhere in the middle, that is when your fitness is supposed to be the highest. So this kind of a scenario where the extremes on both sides are rejected, this is what is known as stabilizing selection. So in this particular case, although selection is operating, you don't really see the trait value changing in any direction, right? And that is supposed to be the most common kind of selection that ends up happening. And the third type is where the fitnesses of individuals in both the tails of the distribution, if the fitnesses of these individuals are much greater and the fitnesses of the individuals at the center of the distribution is much lower, then what happens is that the frequency of these guys come down, the frequency of these guys go up, the frequency of these guys go up. So essentially, from a unimodal distribution, you get to a bimodal distribution of trait values. Now this kind of selection is relatively rare in nature. However, there are a few good examples, and I'll quickly give you one such example. So this is a bird called the black-bellied seed cracker, and there are two morphs of this bird in the natural populations are primarily found in Africa. So you can see that these guys have large beaks and these guys have relatively small beaks. Remember, they're the same population, they're the same species. Okay. So you have these two morphs, and if you look at the distribution of the bill width, then you can clearly see the bimodal nature. So here is one mode and here is another. Okay. Note that within each mode there is a roughly bell-shaped distribution in both of them. Okay, you can almost see a third mode over here, but they are typically here talking about two modes. Very interestingly, this bimodal thing is found only in the context of the lower bill width. If you take the same birds and you measure some other property in them, so for example here they are measuring the tail length, then the same birds show a unimodal distribution. Okay, unimodal distribution means where there is just one peak, bimodal means when there are two peaks. Now, there was this fabulous study, Smith and Girman 2000. What they did was, they took these birds and then so this is the bill width plotted from lower to higher in this direction, and then they actually plotted the performance of these bill widths to show whether the differences in this beak widths are actually having some effect in terms of their ability to do some task or not. So what they showed was that there actually is some difference in terms of the amount of force that these guys can generate, which in turn can affect what kind of seeds they are able to crack. So this is what is plotted over here on the y-axis, and then they took all these birds and then they ended up looking at their fitnesses, and the fitness was in terms of juvenile survivorship, okay, over a period of about 7 to 8 years. What they found when they plotted all these things on the same plot, a 3D plot, what they find is that here it's not one peak, but there are two peaks. And these two peaks are essentially corresponding, the fitness peaks are corresponding to these peaks of the corresponding bill width distribution. So essentially this is one peak where you can see the guys in the middle have the highest fitness. Remember, this is fitness being plotted over here, not frequency. And over here it's not that obvious, but actually you can see that there is one more smaller peak over here. But again, the guys who have like the middle one over here, the middle beak widths, they are the one who have the highest fitness, suggesting that there is divergent selection in this. If you have smallish beaks in general, then you have one fitness distribution, and if you have largish beaks over here, then you have another fitness distribution. So hopefully this species at some point is going to split into two species. Okay, so that was an example of divergent selection. So now we are going to talk about a few properties, a few things that are worthy of watching out in terms of natural selection. So natural selection, it acts on individuals. However, remember, for natural selection to operate, you have to see whether the traits are inheritable or not. So in order to see that, you have to see what is happening in terms of its offspring. Okay, and therefore, although natural selection itself will act on the individual, its effect is going to be seen on the next generation, and that is why its effect is only observable at the level of the population. Why is that? Because remember, individuals die, it's the population which survives over time, and that is why the population is the unit over which microevolutionary changes at the phenotypic level are happening. Secondly, although natural selection is acting on phenotypes, we say evolution is happening only when the allele frequencies are changing. Okay. Now here one might say that that's not what we did in the Darwin's finches cases, because we went from phenotypic distribution to phenotypic distribution. Yes, we did that. And that is under the strong assumption that there is a very good correlation between the genotypic distribution and the phenotypic distribution. If that strong association is not there, then this is going to fail. Technically speaking, the evolution is happening if and only if there is a change at the level of the allele frequency distribution. We very often do a shortcut, make an assumption and check it at the level of the phenotypes, but that's not the technical definition. This I'm going to skip. Selection is always behind. Now, natural selection changes gene frequencies in populations after they are exposed to selection. Natural selection cannot predict in which way things are going. Okay, and the mutations, these mutations are supposed to be pre-existing. Suppose a bacteria gets exposed to antibiotic, then it's not that now antibiotic resistance favoring mutations are going to increase. Antibiotic favoring mutations will happen at a higher rate. It doesn't work that way. Mutations are happening at whatever rate they are supposed to happen. Antibiotic resistant mutations, when there is no antibiotics in the environment, they don't have a fitness benefit and therefore they don't increase by themselves. However, the moment you put antibiotic in the environment, now those individuals which carry antibiotic resistant mutation, their number is going to increase in the population, their frequency is going to go up. But that is because of a selective advantage. That is not because that new mutations which favor antibiotic resistance are coming up at a higher rate. Okay, so mutations are completely not dependent on what fitness, the occurrence of the mutations are not dependent on the fitness values that they are going to accrue. They are random in terms of their fitness values. Now, because selection cannot anticipate future changes, therefore, in that sense, evolution as a process, it's not a future thinking process. It's not a forward thinking process. New mutations arise by whatever is their probability. They do not arise for a purpose. Okay, so evolution is a blind process in that sense. And selection doesn't always improve an organism. Okay, it increases fitness along certain axes, but that doesn't necessarily mean it is leading to progress. And there are many people who end up saying that evolution always leads to greater specialization, greater complexity. Sometimes that happens, but that is by no means something that is an universal fact. So for example, there are several organisms which end up becoming simpler. Okay, so for example, look at parasites. Okay, or look at, for example, cave fishes, when they lose their eyesights. So organisms, when they go into a new environment, they simply do whatever is needed to become more fit in that environment. And if that means losing their specialization, if that means becoming simpler, they do that. And that's what I mean over here, that adaptation doesn't always happen through character gain. It can happen through character gain, it can happen through a loss of a character, or it can happen through the modification of a character to become something else. So for example, you look at the panda's thumb. So you know that we human beings, we have an opposable thumb, which means that the thumb can come opposite to the 4 digits, which is what allows us to form grips. And it is thought that it is because we are able to form grips, that's why we are able to hold things properly, maneuver them, which is what has a major contribution in terms of our ability to use tools. Now it turns out that there is another animal which can form a similar grip, and that is the panda. So the panda has a thumb, okay, which is again opposable, and for all practical purposes it looks just like our, it performs the same function as our thumb. However, if you dissect a panda out, you will find that this thumb is actually not the 5th digit as in us. Okay, so the panda has 5 digits over here, and it has a 6th thing which acts as the thumb, and anatomically speaking, that is essentially the wrist bone, one of the wrist bones, which has become modified to perform the function of a thumb. So this is what I meant by character modification. Right? Now selection, since it operates on pre-existing variation, therefore selection has to make do with whatever is available to it. There is no concept of perfection over here. So, for example, there are lots of people who try to take what is an engineering solution and try to argue that selection is supposed to have made the organization, make the organism perfect, and the perfection is defined as that engineering-wise optimal solution. But that's not how selection works. Selection has to operate on whatever variation is available to it, and then it has to operate within the constraints of what is the nature of the selection force that has been applied, and these two together are going to interact and take the organism wherever it does. There is no guarantee that selection is always going to lead to a quote-unquote perfection. There is no concept of perfection in the context of evolution. And so, for example, just to give you one more example of this is a fish species called Gambusia affinis, and these guys have these things known as gonopodias, which in the males, which is something that ends up attracting the females. So the larger the gonopodia, the longer the more attractive is the male for the female. However, these also have a problem, because these, as you can see, they just stick out like this, and therefore they end up reducing the ability of the male to escape the predators. Now, if you have an environment in which there are no predators like this one, then the selection pressure is primarily on the male's ability to attract the females, and therefore they can evolve longer and longer gonopodias. However, if the same species of fish is made to grow in the presence of the predator, then it has to balance between the ability to attract the female and the size of the gonopodia that allows it to escape the predator. And therefore this tug of war doesn't allow it to evolve gonopodias as large as these guys. So this is what I mean by an interaction between not only what genetic variation is present, but also the nature of the selection. And the nature of the selection itself can lead to a very different kind or a different magnitude of the solution. Now, one major thing over here is that I said that selection only works on pre-existing variation. Now, if that be the case, you can never have a population go beyond the range of variation that is already present in the population. However, if you actually do a directional selection, very often you find that the population goes into trait values which are completely outside the range of the existing population. So this is a very famous example where they were selecting for the average amount of oil in the kernels of corns. And if you look at this yellow band, this is the range in the original population. And as you can see, very quickly, in less than 10 generations, the population mean went beyond the original population range, and then over a few more, about 100 generations or so, it had gone, even the mean had gone far, far, far beyond the original population range. So how is this possible? So it is thought that this happens in two different ways. One is obviously mutation is happening. So as time progresses, mutations come in, and then if there is a positive selection pressure on those mutations, which means if they are being selectively favored, then their numbers are going to increase, and that can take the population beyond the original range. The other thing that happens, this is only in sexually reproducing organisms, is recombination. Remember that during meiosis 1, the chromosomes are getting chopped up and they are essentially rearranging themselves along the sister chromosomes, and therefore the genes are now coming in combinations that did not exist earlier. Now obviously, if let's say two genes are very rare, then the chances of them coming in combination is relatively less. However, if selection pressure is making them more and more frequent in the population, then recombination will also ensure that their chances of coming together in the same background is also higher. So this is what leads to epistatic variation, and essentially recombination ensures that recombination along with selection ensures that newer and newer epistatic variants are created in the population, and this ends up creating a lot of variation on its own. So this is another mechanism by which a population can create new variation and thereby, with the help of selection, go far beyond the original range of the population in terms of the trait distribution. And then of course you have mutation, which can also lead to new traits altogether, traits that didn't exist earlier. Recombination, it's not clear whether it can lead to a similar phenomena or not. So this is more or less about it. This treatment is entirely based on chapter 3 from Freeman and Heron, which I am going to put up as one of the mandatory readings.